Hi everybody, Dr. Mike here. In this video I'm going to take a look at the B vitamins. I'm going to show you how these B vitamins fit into various metabolic processes of the body. Now, the reason why I'm doing this video is because I think lecturers and professors do a pretty poor job of actually explaining how these vitamins fit into various biochemical processes. They like to say vague comments like it's important for metabolism, carbohydrate metabolism, it's important for the nervous system and skin integrity. And it doesn't really mean anything without showing you exactly what they do and how they fit in. Now, I think the best place to begin is a definition of vitamins. Vitamins are essential micronutrients, so we need them for our survival, and we need them in very small doses, and we must get them from our diet. And the reason why, and this is the clincher when it comes to the definition of vitamins, is because we cannot produce them ourselves. Now, there's water-soluble vitamins, which are all the B vitamins and vitamin C, and then there's the fat-soluble vitamins, which is vitamin D, E, K, and A. Now, the fat-soluble vitamins can be stored in fat in the body. So if we have excess of them, we can store them, which means you can OD or overdose and get quite toxic doses of these if you ingest too many. Water-soluble means it's readily dissolvable in our blood and therefore our urine, and therefore we pee out any excess. So it's more difficult to overdose on the water-soluble vitamins. Now you can see when it comes to the B vitamins, we've got B1, 2, 3, 5, 6, 7, 9, and 12. And you're probably thinking, well, what happened to 4 and what happened to 8 and what happened to 10 and 11? So there were those B vitamins, but they got removed because they no longer fit within the definition of a B vitamin. And the main definition being that we can't synthesize it ourselves. And we found that most of those that fit within B4, for example, and B10, we could synthesize ourselves. So they were removed. So these are our water-soluble B vitamins. What we need to do now is talk about their names. So B1 is also known as thiamine. B2 is known as riboflavin. B3 is known as niacin. B5 is known as pantothenic acid. B6 is known as pyridoxine. B7 is known as biotin. B9 is folate. And B12 is cobalamin. So these are our B vitamins. Now the thing is that when we need to talk about what they do, particularly in metabolism. So that vague comment that your lecturer makes or professor makes in that these B vitamins are all important for carbohydrate metabolism, protein metabolism, and fat metabolism is true. It's very true. But how do they fit in? Now the thing is, in order for them to fit in, they don't necessarily work in this form. So they need an active form that they work in. So let's have a look at what the active forms of each are. So thiamine works as thiamine pyrophosphate. Thiamine pyrophosphate. So if you want to know more about how B1 works within the body, Google thiamine pyrophosphate, also written as TPP. Riboflavin produces the flavonoids, and the flavonoids include flavin, adenine, dinucleotide, which is often written as FAD. It can also produce flavin mononucleotide, which is known as FMN. So that's the riboflavin active forms. The niacin active forms produce the nicotinamides, and the nicotinamides include nicotinamide, adenine, dinucleotide, which is NAD. And in actual fact, it works almost active in its oxidized form. Now remember, oxidized means it's lost an electron. Usually things will have electrons and protons, negative things and positive things. When they're an equal number, they balance each other out and there's no charge. But if something's lost an electron, LEO, Leo, lost an electron is oxidation, so it's oxidized. So NAD is functional in its oxidized form because it's lost an electron. Pantothenic acid. Pantothenic acid works basically as coenzyme A. You may have heard that before, often written as CoA. Peroxidine works as pyridoxal phosphate. Pyridoxal phosphate, also known as PLP. Biotin works as biotin. Folate works as tetrahydrofolate. Tetrahydrofolate, known as THF. And cobalamin works as cobalamin. The reason why I'm telling you this is because the way they fit into these biochemical processes is as these forms, and if you want to know more about them, Googling B1 and thiamine, for example, won't give you much info, but these will give you heaps of information. All right, now let's talk about how they fit in. Let's go one by one. But before we begin with that, I need to show you what I've written up here. So I've basically written up, firstly, a summarized form of glycolysis, which is going from glucose to pyruvate. That's glycolysis. And going from pyruvate to acetyl-CoA, and then through this very important cycle known as the Krebs cycle, also known as the tricarboxylic acid cycle, also known as the citric acid cycle. This process produces a whole bunch of what we call NADH. Maybe something to do with that. And FADH2, maybe something to do with that. And they travel to the electron transport chain that ultimately produce a bunch of ATP. All right, so now that we've got this, how do these fit in? All right, B1, thiamine pyrophosphate. It is an important coenzyme which allows for, to begin with, pyruvate to turn to acetyl-CoA, a very important process. This requires an enzyme called pyruvate dehydrogenase, and what TPP does, it plays around with carbon dioxides. So what we have is, let's draw it in another color, let's draw it in green. We've got B1 working at this point in the form of TPP. Now, in addition to that, B1 TPP also works at this point here. Now, I haven't spoken about what is going on with this little side group that's feeding into the Krebs cycle. Alpha-ketobutyrate turns to propionyl-CoA, which turns to methylmalonyl-CoA, which then turns to succinyl-CoA, which can then work in the Krebs cycle. But what is this? Well, this is how you should remember it. You can feed in amino acids into alpha-ketobutyrate. That's important. Amino acids. And you can feed fatty acids into propionyl-CoA. Fatty acids now. So what's this telling you? It's telling you that you can actually bring in non-carbohydrate-based sources into the Krebs cycle. So here's the carbohydrate-based source, glucose, Krebs cycle. Non-carbohydrate-based sources, amino acids and fatty acids into the Krebs cycle to again produce NADH and FADH2, which again helps us produce ATP. So we've got this process. Now, in order for alpha-ketobutyrate to turn into propionyl-CoA, we also need B1 TPP. B1 fits into alpha-ketoglutarate, going into succinyl-CoA. And so you can see that B1 thiamine in the form of TPP is important for pyruvate to acetyl-CoA, alpha-ketoglutarate to succinyl-CoA, and for alpha-ketobutyrate into propionyl-CoA. Now all of them have this CoA snapped onto them. We'll get to that in a sec. Maybe we'll get to that when we talk about pantothenic acid. Next is riboflavin. In the form of flavin-adenine dinucleotide, FAD. Don't worry about FMN at the moment. Let's just focus on FAD. This plays a really important role going from succinate to fumarate. So let's do this in blue. We've got FAD here, and what it does is FAD comes along and produces FADH So this is again B2. And why is this important? What's it doing? Well, simply put, I'll draw it up here quickly, is we have FAD. What FAD does is it steals 2 hydrogen. Now think about it. 2 hydrogen, if you've got a hydrogen, it's got an electron, it's got a proton. So, if it steals 2 hydrogen, it's actually stealing 2 electrons and 2 protons. Now if it does this, what do you think it produces? It produces FADH2, 2 hydrogens, because at the end of the day it's stealing 2 hydrogens. Now, why is this important? I'll get to that in a sec when we talk about nicotinamide adenine dinucleotide. So, that's where it's working here, stealing 2 hydrogens from succinate to form fumarate. Now let's talk about niacin, B3. Nicotinamide adenine dinucleotide in its oxidized form NAD+. It's working importantly here, pyruvate to acetyl-CoA. So we've got B3 working here, and what's it doing? NAD, let's do another colour that you can see a bit better. NAD+, steals just like FAD does, 2 hydrogen and forms NADH. But what it releases is a spare hydrogen. Let's draw it up. NAD plus takes 2 electrons and 2 hydrogens, but one of those hydrogens it spits off. So it throws away a hydrogen. So what it's left with is 2 negative electrons, 1 positive proton, and therefore it produces NADH, only 1 H, but it's gotten rid of that charge because the excess positive charge is balanced out by the excess negative charge here. So at the end of the day, we produce FADH2 and NADH. So we're producing NADH here, we're producing NADH here, so again that's B3, B3, we're doing it here as well, NAD plus, NADH, B3, and we're producing it here as well, NAD plus, NADH, B3, and we're producing it here as well, I know a whole bunch of places, NAD plus to NADH. Alright, so as you can see, 1, 2, 3, 4 NADH are being produced just in the Krebs cycle alone, okay? Or if you're being very strict here, 3 just in the Krebs cycle alone and 1 FADH2. Why is this important? Now I need to show you the importance here. No one really explains it. Everyone says, oh, it does this, but how is this important? So both FADH2 and the NADHs that are produced travel to the electron transport chain. So remember that we've got, so this is happening in, this Krebs cycle is happening in the mitochondria. So we're going to have the mitochondrial membrane here, right? And you're going to have all the various proteins associated with the electron transport chain. Right? All these various proteins. Don't worry about the specifics. NADH, so we've got FADH2, FADH2, and we've got NAD+, which is turned into NADH. What they do is they release their various electrons and their various protons. Okay? So does FADH2. Releases its various electrons and it releases its various protons. Now what happens is the electron transport chain or pump takes the electrons and passes them through. So it takes the electrons and simply passes them across. It's playing hot potato with the electrons. Now, as it plays hot potato with the electrons, it then creates this ability to take the hydrogens and pump them across. And we pump a whole bunch of hydrogens across the membrane. Now, in actual fact, when we pump all these hydrogens across the membrane, we create a gradient. And so these hydrogens, which have now been pumped across the membrane, which now accumulate, there's a gradient where there's a lot of hydrogen and not enough hydrogen. So when these hydrogen then move through, I'll continue this here, this particular channel and diffuse down, it allows for energy, right? Energy is being used here. Well, energy, in actual fact, is being produced because the energy gradient of going from high hydrogen down to low hydrogen allows for us to produce ATP. We can use that and spit out a whole bunch of ATP. So this is called an ATP synthase, and it works by using the hydrogen gradient going down. All right? So the whole point of FADH2 and NADH, which is produced by this process, and importantly because of B2 and B3, is we produce a butt ton of ATP, 32 to 36 ATP molecules for every glucose molecule. All right? So that's super important. So now we've done B3. The next point we need to look at is that of pantothenic acid, which is B5. So this is in the form of coenzyme A. So anytime you see one of these molecules turn into something that has coenzyme A or CoA on the back, that's because of pantothenic acid in the form of CoA. So B5, and I've got to choose a colour now. What colour should I use? I'll do blue. So B5 is going to be working here as well. So going from pyruvate to acetyl-CoA. It's going to be working here, going from alpha-ketoglutarate to succinyl-CoA, B5. It's going to be working here, going from alpha-ketobutyrate, so that's glutarate, alpha-ketobutyrate to propionyl-CoA, B5. But also, pantothenic acid in the form of CoA is important to turn fatty acids into propionyl-CoA, so here as well. Very important. All right, so as you can see, any place in which we've got something turning into a CoA, we're going to have pantothenic acid come along. Next point we need to look at is that of pyridoxine in the form of pyridoxal phosphate, so PLP. What does PLP do? PLP plays an important role when it comes to amino acid production, or at least as an aminotransferase. So it transfers amino groups from amino acids. So let me show you what I mean here. I'm going to do this in pink. So alpha-ketoglutarate can, with the help of various amino acids, like alanine for example, and with the help of B6, which is PLP, what it does is it takes the amine group, so there's going to be an amine group, right, on the side of this, because it's an amino acid, NH3+. It takes that amine group, gives it to PLP, and what it can do is it can produce from alpha-ketoglutarate something really important called glutamate. And as a byproduct in this process, it spits out pyruvate, which is very cool. So from alpha-ketoglutarate, PLP using an amino acid can produce glutamate and pyruvate, and it's reversible as well. That's an important point. Now glutamate, PLP also plays a very important role here in turning glutamate into something called GABA. GABA is an inhibitory neurotransmitter. Again, PLP plays this role, B6. Can turn glutamate into GABA. GABA, inhibitory transmitter. I want you to think about this. You've got excitatory neurotransmitters and inhibitory neurotransmitters. So if somebody is deficient in B6, it means they don't have enough pyridoxal phosphate, which means they cannot convert glutamate to GABA, which means they don't have enough inhibitory neurotransmitters, and one of the symptoms they can manifest is seizures. So that's the importance of B6, and that's where B6 fits in, by glutamate to GABA. It also can actually turn 5-hydroxytryptophan into serotonin and histidine into histamine. Really important. What are we up to now? We're up to B7, biotin. Okay, so where does biotin fit in this process? Biotin actually is important because it transfers carbon dioxides from propionyl-CoA to methylmalonyl-CoA. So this is B7 biotin. Transfers carbon dioxides. Now, this is important, obviously, because amino acids feed into alpha-ketobutyrate, fatty acids feed into propionyl-CoA, and the rate-limiting step here is enough biotin. So if somebody is deficient in biotin, what do they accumulate? They accumulate fatty acids in the blood and amino acids in the blood. They're both acids. They're both organic acids. So they result in something called propionic, because of propionyl, propionic acidemia. Propionic acidemia is an accumulation of organic acids, like fatty acids and amino acids, in the blood because they don't have enough biotin, folate. And the problem that results here is a whole bunch of nervous system disorders. And again, system-wide disorders, skin, so forth. Okay, that's B7. B9 and B12 we should talk about together because they're really important in the process of producing DNA, synthesizing DNA. So let me talk about something which I haven't spoken about yet. And this will bring me back to B1. So hopefully you don't mind. And I may need to wipe this off, so I apologize about that. Glucose can turn to glucose 6-phosphate. But importantly, glucose 6-phosphate can go down another pathway. This pathway, where we turn glucose 6-phosphate into ribose 5-phosphate, is known as the pentose phosphate pathway. Pentose phosphate pathway. And it's a pathway that's important to produce nucleotides, which we know is important for DNA synthesis and RNA synthesis. So, in order to go from glucose 6-phosphate to ribose 5-phosphate, we actually also need TPP. So, TPP comes along, which I did in green before. So, B1 TPP is really important at this process with an enzyme called transhexylase. All right, so transhexylase is playing a role. Or is it transketolase? Sorry, transketolase. Transhexylase is somewhere else. So transketolase and TPP allow for glucose 6-phosphate to start moving down the pentose phosphate pathway. Now what ribose 5-phosphate can do is it can actually produce purines and pyrimidines. Purines and pyrimidines. These are different types of nucleotides, basically, right? So the purines are going to be things like, I'll do another colour here. Purines are going to be adenine and guanine. And pyrimidines are going to be thymine, cytosine, and uracil. And for those of you who know your biochemistry, your nucleotides, you've got A's, C's, G's and T's for DNA, and you've got A's, C's, G's and U's for RNA. That's the adenine, guanine, thymine, cytosine and uracil units, all right? They can obviously be turned into adenosine, guanosine and so forth. All right, so this pentose phosphate pathway produces the nucleotides which are important in DNA and RNA synthesis. What were we talking about? Well, we're talking about B9, folate. Folate in the form of tetrahydrofolate, THF. This will only occur if THF is present. So we need THF here, which again is B9. So what does that tell you? It tells you that folate is important for nucleotide synthesis and therefore DNA and RNA synthesis. This is why pregnant women need to supplement with folate because they've got a child which is growing and developing, and the cell cycle is occurring, and new DNA and RNA are synthesizing. And that's why we need enough folate, because it's important for this process. Now we need to bring in cobalamin B12 because B9 and B12 work together, and this is the reason why. Tetrahydrofolate, its job is to transfer single carbon groups, right? So anything with one carbon. And something that it really likes to transfer that has one carbon is that of methyl. So methyl is a single carbon with 3 hydrogen, all right? It really likes to transfer that. But here's the thing. When THF is floating around in the cell, it will get methylated by amino acids like histidine and serine, for example. So I'm running out of room here, but let me see if I can fix it up. I'm going to have to do it here. So we've got THF, and histidine and serine, for example, can come along and give it a methyl group, CH3. If it does this, it's not functioning. It's not going to do anything. It can't participate in nucleotide synthesis, DNA synthesis. So it's just sitting there. It becomes a sink for THF. Methyl tetrahydrofolate is what it's called. So we need B12. B12 is its best friend. B12 says, don't worry, THF, give me that methyl group, I'll take it off your hands. And so B12 comes along. B12, cobalamin comes along and steals that methyl group off it. It says, I will carry this burden for you. And that's important because it now means that you've got free THF, dropping pens everywhere, you've got free THF that can now participate in DNA synthesis. But what does B12 do? Well, importantly, B12 takes homocysteine, gives it that methyl group to produce methionine. And methionine is very important for a whole range of things in the body. So B12 gives homocysteine the CH3 to produce methionine, which is a whole bunch of stuff in the body. And in doing so, frees up tetrahydrofolate to work in DNA and RNA synthesis, which means both B9 and B12 are important in DNA RNA synthesis. Let's write it down. Both are important in DNA and RNA synthesis. And if you think about it, what cells are most susceptible to this process? Any cell that's going to be rapidly being produced or dividing, like red blood cells, because we make like a kajillion of them every second, are going to be very susceptible. So in order to make or undergo the process of erythropoiesis, the production of new red blood cells, we need both B9 folate and B12 cobalamin. People who lack B12, so here's an important point with B12. Most of the B12 we have is produced by gut bacteria. So it's synthesized by gut bacteria. And that gut bacteria synthesizes the B12 and we need to absorb it, but we can only absorb it if we've got something called intrinsic factor present. Intrinsic factor. Now, intrinsic factor is produced by the stomach. So if somebody has chronic gastritis or damage to their stomach and they don't produce intrinsic factor, it means they can't absorb B12, particularly that from the diet, and they become B12 deficient and their red blood cells aren't being produced, and they can get B12 deficiency-induced anemia called pernicious anemia if it's because of this issue with the intrinsic factor or an issue with not enough B12. So B12, bacteria can produce it and we can get it from our diet. Both B12 and B9 are important for DNA synthesis. This is a lot of stuff, but hopefully I've helped you out now and you know from B1 down to B12 its role and where they fit within metabolism.